A very good morning to everyone. Welcome to the eighth day of UGC MMTC online refresher course on bioscience, bioengineering, and nanotechnology. It's our honor once again to have with us today's first resource person, Professor Shubhit Chandra Dasgupta, Honorary Emeritus Professor of Ramakrishna Mission Vidya Mandir, Belur Math, Howrah. His previous speech on nanotechnological assessment of venom nanotoxicology was truly impactful, and we are delighted to welcome him back. Today, he will enlighten us with his talk on green nanotechnology, the latest innovations and knowledge gap, future perspectives in toxinology. So, without further wasting time, I warmly welcome Professor Dasgupta to deliver his another presentation. Sir, the stage is all yours now.
Thank you, Devolina. I think the slide is visible, no?
Yes, sir.
Yes, sir, visible.
Yes, sir.
Okay. So very good morning to all of you. So today's lectures, as I have already told you, mainly focuses on our lab work, especially my work and involvement in the nanoscience with regards to the venoms and the area of the toxinology. This is rather a new branch in the toxicology or toxinology rather, and our focuses and aim are to reduce the venom toxicity either in the lethality or in the different tissue damage level. So coming back to green nanotechnology, so why it is called green nanotechnology and why this green nanotechnology is involved in the venom research, I will explain one by one. So whenever the term green is used, it refers to the environment friendly objective objects, and the technology means the application of the knowledge for practical purposes. Our work is specially related with the green technology because we identified so many plants in Sundarban area, that plant containing the active agents which neutralizes the venom toxicity, right? So the conventional therapy, as I have told and all of you know, that is an the anti-serum treatment till now in Indian scenario and also in the global scenario, the anti-serum is the only treatment against snake bite envenomation. And in other countries, the poisonous animal like the scorpion, like jellyfish, etc., where the toxin is fatal to the human being, there are the anti-serum against jellyfish venom, against scorpion venom, etc. But in our country, only the venomous creature, the snake venom is predominant, and anti-serum is usually available in our country, which is prepared against the four venoms, big four snakes in India. So one is crate, another is cobra, another is viper, and another is saw-scaled viper. All these venom are combined and that push into the horse, and against that the antibody is produced inside the horse, and ultimately from where this anti-serum has been prepared and available in Indian market, one vial cost approximately 1500 to 2500 depending upon the company made. But nowadays this treatment has been changed abruptly, and the involvement of this nanoscience make this possible. And I'll just share some of the idea what are the present day status of this snake bite envenomation involving the nanoscience, and the involvement of our laboratory, and what we actually focuses involving the nanoscience to reduce the toxicity of the venom using the plant extract. So this is green nanotechnology, is a study how the nanotechnology can benefit the environment, such as by using less energy during the manufacturing processes, the ability to recycle the products after use, and using eco-friendly material. So the principle of green chemistry is that less hazardous chemical synthesis, right? Synthetic methods should avoid using of generating substances toxic to human or to the environment. Our target is to reduce the toxicity by applying the nanoscience and designing safer chemical, that is chemical products should be designed to achieve their desired function while being as a non-toxic as possible. Totally it is able to reduce the toxicity is difficult, but the fold of the neutralization, fold of the protections is a very important, right? So there are a lot of tools have been used in the green technology. One is green starting material, green reagent, green reactions, green chemical products. We use the green chemical products and the green methodologies too. Now, research in the field of cellular and the animal interactions of the nanoparticle is vital owing to its molecular chase game, right, which is plays within the system. This opens a new door to the use of nanomaterial in the drug delivery and the targeted therapy. The modern trend in the snake venom research include utilization of the nanotechnology. Research in nanotechnology and the snake venom or the toxins has gained two different dimensions. One, use of nanotechnology in the development of snake venom antidote, and number two, application of nanotechnology in the development of drug clue against pathophysiology like arthritis, cancer, HIV, whatever the disease. Till date, anti-snake venom antiserum, that is ASVS, has been the only clinically used antidote against snake envenomation, developed in 1894 at Pasteur Institute, Paris, by Dr. Calmet. He is the father of this anti-snake venom antiserum, but it's carefully about the year 1894. The limitation, the cost factor, the shelf life, no protection against local effect, hemorrhage, and organ toxicity. That is the limitation of that anti-serum, because if you think properly, in Indian system, the main disadvantage is to that the identification of the particular species, which species actually bite the human being, right? And in West Bengal, if you consider about the West Bengal, uh, actually the bite occurs maximum in the Sundarban area. When I personally visited along with Dr. Gomes, we identified several plants that is used by the ojas, though oja are not the scientifically is acceptable, but the local villagers of the Sundarban, they rely on the ojas. They usually bring some plant and use the extract for that victims, so that the victim can regain some of the senses and reduces the inflammation or the other pathophysiological symptoms. So we asked the oja, several ojas we have interrogated, and they said there are two or three plants they use. So we identified and collected that plants and tried to establish or isolated the compound from the plant. We proved that yes, that plant containing active ingredients that neutralizes this snake venom toxicity. So if you make an experimental trial, in one set you applied the antiserum that is available in the market, and the second trial you applied the plant extract into the rat model that is already injected with the venoms. We observed that the lethality, the terms of LD50, is a much more improved if you use the plant extract. Immediately, we go for a isolation of the active component from the plant, and you know that to isolate the compound from the plant is a very challenge. So still that we isolate the compound, we characterize that compound chemically. I will show later on. And that chemically compound, after chemical analysis, we purchase that chemical from the Sigma. It shows that the same chemical also protect the snake venom induced victims. So then we have a standardized method to isolate the compound in our laboratory. So naturally, what we do, we actually try to tag the nanoparticle with these active compound isolated from the plant and applied in the animal model to check whether the LD50 protection or MLD protection offered by the plant extract has been increased or not. This is our primary goal with the nanoparticles tagged with this plant extract. I'll show how we will proceed and how we will characterize all the events. Finally, there are a lot of we tested for the lot of snakes, and finally we are able to characterize in details also the histological alterations that is carried out by the snake venom. We found there is a protection and the histological damage by the nanoparticle. So the involvement of the nanoparticle in the venom research is very rather new. Recently, a group of scientists, as I have told in my last lectures, that recently a group of young scientists has been joined in the Indian Institute of Science, Bangalore. They have tried to prepare this nanoparticle induced the venom's antiserum. So I think within a couple of years that nanoparticle or the involvement of the nanoscience regarding snake venom envenomation has been possible. The modern trend in snake venom research includes the utilization of the nanotechnology, right? Research in nanotechnology and the snake venom toxin gain two different dimensions. And till date, anti-snake venom serum has been only the clinically used antidote against envenomation. The limitation is a much more important factor because majority person, the person is unable to tell whether actually the snake bite or not. They said that yes, the snake bite is there. Naturally, what happened after admitted to the hospital, doctor without checking, they administered the antiserum. So vigorous antiserum reaction have been started inside the body. There is an anaphylaxis, and the patient ultimately died. But that death is due to the antiserum sickness, not due to the venom. So it is very difficult to identify whether actually bite occurs or not, and if bite occurs, how much amount of venom is given by the snake inside the body. That is still there is a very big problem, but unfortunately we have tried because if you consider the global scenario, there is a bedside diagnostic kit by which one can easily identify whether the snake bite occurs, and if the bite occurs, which snakes actually bite. This is very important. And bedside diagnostic kit is already available in UK. The pioneer scientist is Threkston, and who identified and established the bedside diagnostic kit. We have tried but failed because it requires a lot of grant to prepare that bedside diagnostic kit. The basic principle is nothing but a SRID, that is single radial immune diffusion. So if the plate is available, if you take the one drop of blood and put inside the plate, so the blood act as an antigen, and if there is an antibody inside the plate, that immediately gives a halo ring from where you can quantify how much venom has been introduced inside the body, and whether the actually the snake bite occurs or not. But we have failed to produce that bedside diagnostic kit. Naturally, worldwide scientists are looking for a better anti-snake antidote. The herbal resource of the world having anti-snake venom potential have been known for a long time, even in the Susrut Charok. For that reason, the herbal plant use has been very common in Indian history. Folk and traditional medicinal system mention about different herbs active against snake venom. The first,
Excuse me, sir.
uh,
Sir,
yes,
sir, slides are not
no, no, no,
moving.
I know. So,
And one question I have, sir. You said
yes,
that snake bites where it couldn't be identified that whether the snake has bitten the person or not. As
right.
we know that if snake bites, if it is a venomous snake, then there should be fang marks. And if it
No,
is
no.
this
If,
venomous
if
snake, there will be multiple
yes,
fang marks.
yes. But what you told, that fang marks and identification is for the knowledgeable person, considered about the layman reside in the Sundarban. They
Okay.
do not know what is fang. They do not know what is the bite by the fang. They do not know what is the distinguished character between a poisonous and the non-poisonous snake. So how they can identify the fang is by bite by the poisonous snakes? Considered about the layman, because in town and in urban city, bite is very limited.
Limited. So
So
they
I
can only see that there are marks, and they consider it that
yes,
snake has,
yes, that
and
is
they.
possible for our people who
Hmm.
knows what is the actual the bite sign. But considered about the remote village of the Sundarban.
Excuse me. Excuse
Yes,
me.
yes.
Dear participants, you have question hour. Please don't interrupt the speaker session. Okay, please, please.
Okay. Okay, sir.
Okay,
Sorry.
thank
So,
you, Professor Dattu.
yes. So, what I says is that folk and the traditional medicine system mention about the different herbs active against the snake venom. And the first evaluation of the herb as a snake venom antidote was done by Knowles. Is a very old. He first reported, yes, there is a possibility of the plant that has been used against the snake venom bite as a substitute of the antisedum, and screened several plant constituent used by the traditional practitioners. And if you go for these homeopathic doctors, you can even find there are a lot of homeopathic drug prepared from the snake venom, that is Vipera 30, Naja 30. Lot of homeopathic drug is there. So our target is to prepare this in the context of the snake bite. Nanoparticle have shown promising result in the development of more efficient therapies, right? So snake venom, I have here focused only the snake venom, but don't think that this concept is not applicable to other toxin also, because and throughout the world there are a lot of animals which contain the toxins. But the here we have very much keen to solve the problem, because the mortality rate due to the snake bite in Indian scenario is quite high. That data we have collected from the Indian Council of Medical Research. But other country, the jellyfish poisoning is a big problem. Other country, the scorpion bite is a big problem. In other country, cone shell, that is a mollusk, is a very problem. So depending upon the nature of the mortality, that basic principle is applied in the different field of the toxinology. So in the context of the snake bite, nanoparticle have shown promising result in the development of more efficient therapies. The encapsulation of the snake venom in polymeric nanoparticle, as an example, is well studied due to its biodispensability. That means the effective physiological alteration inside the body, and the selectivity for the treatment, as well as the decrease in the systemic toxicity. I have already told our target how this toxicity can be diminished, right? This is our focal point. So if you consider this snake, they have the so many I have identified, that is from the literature, that is not our work. They have identified so many basic key components which are the cause for the mortality. So the number one is C type of lectin, and its function is carbohydrate. A calcium dependent carbohydrate protein causes homeostasis, modulation, blood coagulation. Example for the viper is a hemotoxic venom. This is 3FX, means three finger toxins. If you isolate the toxin and characterize through XRD, you can find the nature of the toxins, crystallographic analysis, so that the better drug binding can be possible. Small protein toxin with a structural motif. This is PLA2, is a dangerous enzyme present in the all snake venom. This is phospholipase A2 enzyme that hydrolyzes phospholipid, causing myotoxicity and inflammation. It is almost equivalent a cytotoxic agent. This is SBMP, that is snake venom metalloproteases, another group of enzyme that is zinc dependent proteolytic enzyme, causes hemorrhage, coagulation, and inhibition. This is snake venom serine protease. The scientists have identified so many groups of the enzyme, which is very fatal. and causes serious a series of pathophysiological alteration. That is serine-dependent proteolytic enzyme causes coagulation inhibition. This is LAO, that is L-amino oxidase, is the oxidoreductase enzyme, causes cytotoxicity. And this is the disintegrate naturally the small cysteine protein causing the inhibition of platelet aggregates. So these are the, in general, the broad avenue, the broad path through which the snake venom act and causes the mortality. So the representative of snake venom characterization, there are a lot of method, and before I go through our work, I must acknowledge the lot of the other scientists beside the zoologist and the physiologist. I am from a zoology background. My mentor is from physiology background, but beside that we took help the other chemists, like from the biochemistry department. Professor Dashgupta is there in the biochemistry department. We took help from the Indian Association of Cultivation Science, Department of Biological Chemistry. We took help from the Indian Institute of Chemical Biology for the detailed chemical analysis, because we are rather handicapped because we are the life science student, not very sure to characterize how this particle can be assessed. We just carried out the biological testing after binding and after preparation of the nanoparticle, characterization of the nanoparticle. Then we tested whether this nanoparticle can reduce the toxicity and alterations can be diminished in the other tissue level. So this snake venom particle isolation and the characterization that have done we in inside our laboratory, we have purified the exact lethal component through ion exchange chromatography. Then it is a physical, physiochemical characterization, DEXA scan. There are a lot of methods by which you can nanoparticle can be characterized. I will show you later on. And then testing of the biological activities, and there are a lot of testing method. Mainly the viper, if you consider about the viper, it is basically a hemotoxic venom. That means the basic component and the toxins can act on the blood coagulation system. Naturally, we tested on the blood coagulation system. Some venom are acting as an cytotoxic. That means we are testing about the different bacterial strain in collaboration with the microbiology department. So representation of the snake venom, characterization and evaluation for the biological uses. So there are so many paths for this formation of the nanoparticle tagged venom particle. Here we have only used the gold nanoparticle. Out of this broad domain, we just carried out with a C. Occasionally, the gold may be replaced by the silver, but it is still pending to work out with a polymeric nanoparticle. But I'll show you some paper, that recent paper, where the scientists can try to prepare the polymeric nanoparticle to combat the lethality, to reduce the mortality. Then it is a liposome technology. That work is at present carried out by the Indian Institute of Science. And there is a silica nanoparticle, and the last one is a functionalized nanoparticle. So there are altogether 5 branches through which this area can be ventured. The use of the nanoparticle is based on the incorporation of the proteins derived from the snakes. That number 1 is polymeric nanoparticle, number B is a liposome nanoparticle, C is a gold nanoparticle, D is a mesoporous silica null particle. It is a non-toxic schematic representation showing that nanoparticles, regardless of the nature, can be modified, thus improving their physical, chemical, and the biological properties. So there is an animal toxins within the nanoparticles, right? Here are schematical presentations of a cobra. And I have, this slide is rather a representation from a toxinologist. That is, you may use the bee venom because bees nowadays also the wandering agents, because you know the disease multiple sclerosis is a dangerous disease, but the only drug that is available from the bee venom. So scorpion venom is a terrible anti-cancer, also anti-osteoporosis. So all these venoms can be used for the therapeutic uses with the help of the nanoparticles, right? So this is a very symbolic slide of the involvement of these nanoparticles. Now again here it is a different toxicological effect, that is cytotoxicity, neurotoxicity, hemorrhage, myotoxicity, endothelial cell adhesion, and pharmacological effects, anti-tumoral, anti-angiogenic, antimicrobial, cellular signaling, and neuroreceptor recognition. So there are a lot of paths. I have mentioned only few because I have mentioned this based on the recent pathway or the recent research that have been carried out with the using nanoscience in the venom research. So they have identified, again, scientists have been identified some of the group I have already mentioned. But if you look carefully, there is some new component addition, that is waprin family, a group of family that is also important. Serine I have mentioned, tripeptidase, three-finger toxin, cephalotoxin, that is also a new bradykinin potential peptide. As you know, the bradykinin is a vasoactive agent. It causes several pathophysiological alterations. The scientists identified the bradykinin involved toxicity inside the living organism. So the scientists pay attention for the tagging of the nanoparticle with the bradykinin potentiating peptides, the metalloproteases, phospholipase, natriuretic peptides, that is NP, disintegrin, crotamine, and Wolgerin. These are the other compound that had been identified, which has been tagged with a nanoparticle to reduce the biological effect. So again, it is a venom peptides. Similar slide. There are a lot of other possibilities of the nanoparticle. You find that is a nanocapsule, carbon nanotube. If you search in the Google, the carbon nanotube and the venom toxicity, you find quite a good number of paper, because throughout the world the scientists are trying. So application of the different form of the nanoparticles, by which particle, which nano particle conjugate can reduce and effective most successfully, right? So therapeutic use, that means anti-hypertensive, anti-cancer, anti-thrombotic, analgesic, anti-leukemia, platelet, anti-platelet, anti-nociceptive, and anti-arthritic, and the myotoxic. So the venom peptide formulated by using nanoparticles for the different therapeutic uses. Now, these are the at a glance our work, the laboratory. From our laboratory, I have participated only three or four, but so that's a very interesting. Go for the title of the paper, then I'll explain one by one. The Daboyer Russeli, that is a common viper, and the Naja cautia, venom neutralizes by the lupial acetate isolated from the root extract of Indian Sassafila hemidesmus indicus. The plant is Hemidesmus indicus. This is known as a Sharpogandha, another one is Sharpogandha, another is Ashwagandha. So we have identified the compound, characterized the compound, which is nothing but a lupial acetate, and that lupial acetate is equivalently very strong, combat the neutralization of this snake venom from our laboratory. Then viper and the cobra, venom neutralization by beta cytosterol and stigmasterol isolated from the root extract of the Pluchia indica. All these plants that we have identified with the able of the our visit to the remote village of the Sundarban, we interrogated the different ojas, and they said these are the plants, but they are unable to identify the nature of the plant. So we collected that plant, bring back to the Kolkata, take help from the Bioscience College Botany Department, also from the Botanical Survey of India. They identify the nature of the plant. Then we isolate the compound, and the compound is like this compound, beta cytosterol or stigmasterol, and that can also effectively neutralize the venom and the cobra venom. Then cobra venom neutralization by the gold nanoparticle by the 2-hydroxy-4-methoxybenzoic acid that I have already mentioned. That work I have partly carried out, tagged with this research team. I'll explain one by one how we can achieve this compound and how this neutralization process being carried out. Next is protection against osteoarthritis in experimental animal by nanogold conjugated snake venom protein toxin, gold nanoparticle from Naja Kauthia venom. Again, this work has been also involved myself and Anjan Kumar Dasgupta. He is a professor of Department of Biochemistry. He actually guided us terribly for the formation of this, how this nanoparticle has been produced inside the laboratory, the characterization of the nanoparticle, and then we effectively use this venom conjugated nanoparticle in different experimental trial, the pathophysiological trial. And the last one is the adjuvant effect and the antiserum action potentiation by a herbal compound, the 2,4-hydroxymethoxybenzoic acid. The previous compound, we saw that that compound, plant compound, is very effective as an adjuvant effect and antiserum action. What I have already mentioned, if you inject this 2,4-hydroxy-4-methoxybenzoic acid challenge with that venom, this mice will be very feel comfortably, the neutralization fold will increase, and again the plant extract known as a sarsapilla again to the hemidesmus indica. So these are the total combined work from our laboratory. Then I'll explain one by one. This is a very current from the Down to Earth magazine. It is published in 31st October 2025. It is a very popular and common magazine. So that magazine, in wildlife biodiversity section, they cover a new antivenom discovered by researcher using nanobodies promises to be a game changer against snake bite. What I have mentioned in my previous lecture, also in the first half of my lectures, that a new antivenom therapy has been opened. That formation has been opened in front of our country. The use of the nanobodies instead of the conventional antiserum treatment, and which plays in a very good promising effect, and they said it is a game changer against the snake bite antibody. And last line, the clinical trial could begin in 1 or 2 years. That I have mentioned, within 2 or 3 years, one can expect that instead of conventional antiserum, the nanoparticle conjugated or the nanobodies against snake bite envenomation can be available in our country, so that a large amount of people can save life, because the mortality is a very higher, to be ready within 3 to 4 years. Now the that papers I have already shown in my first lectures, that is nanobody based recombinant antivenom for cobra, mamba, and wrinkles venom. This is a all the snakes are for from the Africa. This is also a current paper in 2025, 22nd September. But that papers opens a new dimension, because each year snakebite envenomation claims thousands of lives and causes severe injury to victims across the sub-Saharan Africa, many of whom depend on the antivenom derived from the animal plasma as the sole treatment option. Traditional antivenoms are very expensive, can cause adverse immunological reaction, that is serum reaction, offer limited efficacy, local tissue damage, and are often ineffective against all medically relevant snake species, because the antiserum in Indian scenario only four, but beside four there are a lot of snake are there. I have mentioned big four. One is viper, another is cobra. Cobra means it is a naja naja, not naja kautia. Another is a Bangiurus cerulus, that is common known as kalaj, very common in Sundarban, and another is a saw-scaled viper, that is Ichthys carinatus. These big four venom are been cocktailed, and against that antiserum is available in our country. But considered about the bite of the banded crate, Bungiurus fasciatus, it is a very sluggish, yellow and black alterations. It is a very sluggish, bite is very uncommon, but if it bite, then instant death, right? So there is no antivenom against that banded crate. There is no antivenom against this dangerous king cobra, that is Ophiophagus sana. There is no bite against Naja kautia, right? I have mentioned about the cobra, that is commonly known as gokro, but no antiserum against the Keute, that is Naja kautia. At present, I am working on the Naja kautia venom. So it is a very deadliest snake. There is no antivenom. So considering about this paper, though the paper effect and the research is considering about the all the big snakes in the Sahara and African context, I have personally saw the mamba when I visited Masai Mara and Serengeti. It's a tremendous and very fast movement, is very fast, and everybody is scared by looking the mamba. Is a dark black and very thin and sharp, and also is a very common in African country. One is there is Naja something species, is I forget the name, but it is a spitting cobra. They cannot bite, they can just spit the venom targeting the eye of the victim. And if by any chance that the venom can goes inside the eye, it is causes immediate blindness. So the lot of research have been working out. There is no antivenom against spitting cobra. So scientists are trying their best to produce the nanocapsule, nanobody based formulation for the protection of the spitting cobra. And most interestingly, this paper is the first paper who highlights this. Instead of the horse, there are other animals that can be equally important for the productions of the antiserum. So there is an urgent unmet medical need for the innovation of the snakebite envenomation therapy. However, developing broad spectrum treatment is a highly challenging owing to the vast diversity of the venomous snakes and the complex variable composition of their venom. And this papers actually immunizes the all these three venom, that is cobra means it is basically the African cobra. It is a Naja nigrocolis, I remember. Naja nigrocolis, mamba, and the wrinkles, these are the very deadliest snakes in the African context. And they have prepared a phage displaced library, that means involvement of the virus, this phage displacement library, and identified high affinity broadly neutralizing nanobodies. And they use to prepare the antiserum by using the animal what we called as a llama, right? There is a camel, the short camel, llama, and llama equivalent animal that is known as the alpaca. ALPACA. A new animal has been standardized for the productions of the antivenom because in African context, the availability of the horse is really a problem. So they tried to prepare the antivenom, compare with this antivenom prepared in the alpaca and the llama, and basically they use the phage displacement library. So the broad picture is like this. So they have shown this total Africa, the mamba species, the four species are there. That is cobra, that is 13 species. Just remember, there are 13 varieties of the cobra. And the wrinkles, that is a very new snakes. That is a wrinkle which is a highly poisonous. And considering about the data, I have taken the slide from that paper. The paper is like this paper. So I have taken this slide from there. And you just look this number of envenomation. It is 3 lakh to 4 lakh 20,000. It's not a matter of joke. And the death is 7,000 to 30,000. And amputation is less than is 10,000. So amputation means removal of hand and the leg. So they have worked out in details that papers. They said there is a membrane toxin is there, and you found that is the PLA2 is there, the phospholipase is there, the cytotoxin is there, and they usually causes the convulsions by blocking the sodium potassium channel. And in the B figure you find there is an the llama 18 species, the generation of the llama, and that antibody can be procured in the viral systems, phage, and that phage can be used as an antivirus, and that produces the toxins. If you go for the legend, in especially the B, the schematic overview of the pipeline, all these are in pipeline because the first slide shows that within a 3 to 4 years, I think this the standardized method comes into the pictures, and globally the schematic overview of the pipeline used for the development and experimental recombinant antivenom. An alpaca and a llama were immunized with a mixture of the whole venom from 18 elapid snakes to generate immune VHH displaying phage library for the discovering VHHs with broad cross reactivity and high affinity. That is an antiserum tagged. This antiserum tag characterized in vitro and in vivo with a 8 antiserum chosen and combined with an experimental recombinant antivenom. Visual representation of the pathology of the included major toxin family. There are a lot of toxin family. That is, one is cytotoxin, one is membrane toxin, and that can be analyzed with the protein data bank analysis. So this is overall of that paper summary because it is a very recent paper, and the scientists are now and group of scientists have been working. Look, the author how many authors is there. So throughout the world, the scientists are trying to prepare a nanobody based venom combination for the sake of the life of the human being. So the nanotoxicity of the snake venom is mainly covering the as an extracellular vehicle. So snake venom containing extracellular vehicle that act as a natural nanocarrier as a toxin. These and extracellular vesicle can amplify the venom's toxicity by using interfering with physiological process like blood clotting and cellular energy metabolism, enzymic activity. I have mentioned all the different types of enzyme, that is L-amino oxidase, phospholipase A2. There are a lot of particles. So nanoparticle can interact with the venom enzyme like L-amino oxidase, which can lead the oxidative stress and cell death through apoptosis or necrosis, and the cytotoxin. The nanoscale interaction of the cytotoxin with the cell membrane causes alter their organization and function, leading to damage like hemolysis and necrosis. Now, this is again another very current papers. Look the paper's name and the journals. It is a journal of drug delivery science and the technology by the Elsevier's, and it is a very current paper. Look in the top. This is 2025 in the month of the October. This is a review article that means beyond the peril of the envenomation, a nanotechnological approach for the therapeutic venom delivery. So there are a lot of papers, and within 2000, if you go for this publication in 2025, making a caption that venom nanoparticle and the antiserum, you are astonished. You get at least 10 to 15 papers in 2025 where everybody mentioned the dimension and the angle of the treatment. That has been rapid change. Instead of conventional therapy, they all suggested the possible pathway for this development of the nano bots or gold nanoparticle or nanotechnological approaches or nanocarbon tube against this venom, against the envenomation. So this paper actually highlighted the considerable increase in case of the disease that are difficult to treat, highlighting the need to implement new therapeutic alternatives. Active component is with a therapeutic potential were identified as snake venom with a proven biological activity. In Brazil, a country known for its diverse fauna, because there are a lot of variety of species, including the snake, there exists a wide variety of venomous animal, including snake, scorpion, and bees. I have already mentioned this. In Brazil, the most deadliest scorpion is that Titias cerulutus. If one stuck is there, instant bite. So it is a highly venomous, and the venom LD50 is more than the Vipera raceli. In our India, there is not so much venomous scorpion is there, with a mild envenomation takes place. But in Brazil, there are at least 8 to 9 venomous scorpion are there. So they have mentioned about the snake, scorpion, and the bees also. The venom of these animal present therapeutic potential due to the presence of the biomolecule with properties such as an antivenom, anti-cancer, anti-inflammatory effects. However, these biomolecules are easily degraded when not adequately formulated. Nanocarrier formulation have been considered promising for the delivery of the therapeutic bioactive molecules, particularly genes and the proteins. These nanosystems have been widely used in different areas like food, pharmaceutical, cosmetic industry. In pharmacological therapy, the use of nanosystem can provide stability, reduced toxicity, increased half-life, protection from degradation or premature inactivation of the therapeutic agent, enhancing the overall efficacy of the treatment. And different types of biodegradable delivery nanosystems such as a liposome that I have already mentioned. So many angles the scientists are trying, such as liposome, such as polymeric nanoparticle, such as lipid nanoparticle, have shown a high capacity for loading component present in the animal venom, proving to be a very useful tool for obtaining many new pharmacological applications. So what I have already shown, the different angle of the nano technology. So we have just worked for the tagging with a gold nanoparticle. I do not have still any expertise with this liposome or any other particle. But it's fact, it's true that the scientists are trying with the different form of the nanoparticle, and it's very difficult to say right now which nanoparticle show the most effective against the snake venom envenomation, right? So the different angle, the different scientists are working. So the next slide is shows this another a good papers for the PLOS. This is also a very current. So the research article shows that engineered nanoparticle bind elapid snake venom toxin and inhibit venom-induced dermonecrosis. Some of the snake causes huge necrosis inside the skin, right? So the scientists are targeting to reduce the necrosis, and it is a dermonecrosis. And this is a perhaps a bit old paper, 2018, and this mainly focuses. If you go for the abstract, you find this enzymes is phospholipase A2, because almost all snake containing this PLA2, which is a very dangerous. And this basic pharmacological action of the PLA2, it is a membrane toxin. It dissolutes the membrane, causes cytotoxicity, causes necrosis. So the scientists are very curious about to reduce the dermonecrosis with the tagging of the nanoparticle with the different lipid venoms. This is our work. Here we can start from our work. This is the first paper, very old, is 1994. The isolation and the purification and the partial characterizations of the viper venom inhibiting factor from the root extract of the Indian medicinal plant Sarsaparilla. I have already mentioned in early 1992, 93, 94, we extensively visited different rural villages of the Sundarban to witness what are the fate of the people after the snake bite. We have a snake charmer there. He informed us, yes, there is a bite of the snake. Immediately we rushed to that spot in the canning. There is a launch and reach into the Sundarban area. And we found that there are a lot of Ojha, at least 2 or 3 person who claimed there as an Ojha. They use different types of extract, liquid extract, into the mouth of the person. The person is unconscious, is a drowsiness. Then after that, occasionally the people respond, occasionally the people do not respond. Then after quite a long time, period of time, then we interrogate the Oja and we ask them what are the extract, what they use for this the patient that reported to bite with a either viper or it is a Bungiura cellulose. They said there are so many plants. From there, we identified one plant known as the Sarsaparilla, Hemidesmus indicus, and that plant plays a very significant role. And this is the first author whose PhD thesis is there. Now he is a professor in Jamia Millia Islamia University. Is a pioneer work. He identified that yes, there is some compound present in the root extract of that plant, and that extract is very much effective to neutralize the venom of the viper, right? That is a WRSLE. So it is clear that's because the viper is a terrible hemotoxic. If you inject the mice some amount of venom into the mice through its tail, after 5 or 10 minutes, if you pierce the retroorbital complex inside the eye, the mice bleed to death. They're unable to clot the blood because an anti-clotting factor is there, and terrible anti-cancer, terrible hemotoxic is there. So that hemotoxicity can be challenged with this root extract, and we found there is a effective protection by this plant. Next, this again, this is almost similar time, 1998. The same group then worked out the adjuvant effect and antiserum action potentiation by the herbal compound. Then what they carried out with this compound, we instead of using normal adjuvant, that is fried complete adjuvant, here we use to prepare of the antiserum locally. So instead of fried complete adjuvant, we use this compound, chemical compound, 2-hydroxy-4-methoxybenzoic acid. The similar result, the adjuvant effect and the antiserum-like action potentiation with this 2-hydroxy-4-oxybenzoic acid. So this is the first preliminary work by our laboratory, by our mentor, Professor Gomes, in 1998. From that, we have started the nano particle conjugation concept. So our next paper, this is a very good paper published in 2016, application of the gold nanoparticle conjugate with the 2-hydroxy 4-methoxybenzoic acid from the Hemidesmus indicus root, enhancing neutralizing snake venom activity. So with this compound, that compound which we have isolated from this plant extract, is now being used for the different types of the nanoparticle based compositions. So what is that? So the snake venom actually has been caused an innumerable death worldwide. An anti-snake venom antiserum is the only treatment, as I already mentioned. Several herbal antagonists active against snake venom has been identified. Among that, 2-hydroxy 4-methoxybenzoic acid we have isolated from the root extract from the Indian medicinal plant. And nanotechnology is an upcoming technology which has been tremendous potential in biomedical research. The nanoparticle tagged natural biomolecule, including venom toxin, are of interest because of the fact that nanoparticles have unusual properties that can be used to improve the pharmacological and therapeutic properties. And our study was an attempt to conjugate the herbal component with a gold nanoparticle to investigate whether the gold nanoparticle conjugation could improve the snake venom neutralization properties of Hemidesmus indicus plant extract. Gold nanoparticle tagging with HMBA was done by the adsorption method, physiochemical characterizations done by the DLS, TAME, AFM, FTIR, and fluorescence microscopy. So, atomic force microscope is a good method to characterize this nanoparticle. Also, the transmission electron microscope, also FTIR, DEXA scan, and also the fluorescence microscopy. The paper shows there is a adsorption method by which the gold particle has been tagged with this nanoparticle, this this absorption method. Then it is characterized by the transmission electron microscope. This particle size and shape is very important. Then it is put with a dynamic light scattering to calculate the zeta potential. One thing I have clearly mentioned, that portion has been absolutely carried out by a chemistry people, because being a zoologist and physiologist, it is very difficult to identify and to calculate the zeta potential of a chemical compound. So before carrying out the science with a nanoparticles, you must have a good collaboration with the chemistry people. Otherwise it is very difficult to characterize that nanoparticles. A good characterized nanoparticle can be only able to neutralize this the toxicity. So next slide shows, this is the atomic force microscopy. This from this nanoscience center, Calcutta University have a nanoscience center from where we have extensively carried out this atomic force structures, and that structures ultimately shows the nature of the nanoparticles, this the minimum. And then we have analyzed this nanoparticle sources to different physiological combinations. This is FTR analysis. The analysis shows this, in which wavelength this nanoparticle can be best absorb the wavelength. And then finally we can carry it out, this is a fluorescence activity of this nanoparticle. Then we come to this biological activity, effect of snake and the venom induced paw edema and protection by this the nanoparticle in male albino mice. This paw edema can be easily carried out with the injection of the carrageenan powder, simple yeast. If you inject the yeast in the hind paw, the paw will be swelled. You measure the diameter through caliper. Then you inject the nanoparticle induced the particle induced venom into the experimental animal. Then you clearly observe that this nanoparticle tagged venom reduces the paw edema. Here it is so the dermonecrosis and the protection of the gold nanoparticles. So there is a clearly the group B. Group A is a venom treated necrotic spot. This is a necrotic spot If you keep time, if you inject with a viper, this is necrotic spot. If you inject with the viper, there is a more sharp area of this necrotic spot. This is a spinal cord of the rat or mice. So after removal of the skin, you can identify the dermonecrotic spot. Then subsequently, the C is a venom plus tagged nanoparticle that can reduces the dermonecrotic spot. So here in this way, we can analyze the nanotoxic particle can reduces the venom toxicity. So next phase of work our is that the Naja Kautia, the protections of the gold nanoparticles, and that can induces the AKT and mTOR inactivation pathway. As you know, this pathway is mainly responsible for the autophagic reaction and mainly the apoptotic induced cell death in the leukemia cell line, because almost all snake venom are usually capable of the to neutralize the cancer cell. At present, I am working with this Naja Kautia venom, especially for the skin structure, because one paper from the IIT recently, one paper published from the IIT Kharagpur, that the snake skin causes something unusual orientation of the skin so that the bacteria cannot enter through the snake. There is no chance of the infected skin by the any bacteria. So they observed the scientists. So what they have done, as because they are the engineer, that department, that work has been carried out by the mechanical department of IIT Kharagpur, published in 2024. They showed that the snake skin, again this Najakautia, the snake skin causes some beautiful orientation where there is no infiltration of the bacteria. So that is very unique. So what we have tried, we then carried out this extract the skin and undergo the X-ray diffraction analysis. We found that the collagen, which is usually a triple helix structure, and there is a definite orientation of the collagen in a human being, as because all of you have know the collagen structure. But in case of the Naja skin or Naja Kautia skin, the collagen structure is something peculiar. What we observed, that is a very recent study, that is still unpublished data I have carried out. So this XRD analysis revealed that the collagen structure of the Naja Kautia skin is not the book copy structure what we have already did, triple helical twisted structure. It is not in that form. So orientation is something different. As a result, there is no infiltration of the bacteria. So next work I have done with the skin, we have checked the antimicrobial activity with a different strain of the bacteria. Yes, it is positive. The no bacteria can infected the skin. So up to that, we have I have just finished. Then it will still continue. So I am interested about this side because there's an anti-cancer. It already proved positive in the HeLa cell and the breast cancer cell. So our work suggested yes, that is a positive. If the gold particle tagged, it can interact with the signaling pathway and also the apoptotic pathway. So in another paper is also there. So the nano gold conjugation anti-arthritic potential. Here it is the previous paper with like anti-cancer leukemia. Here it is an anti-arthritic potential. It's a very easy to create arthritis in the animal model. If you took the rat and mice and inject the prednisolone injection regularly for a 1 week, the mice developed arthritis. It can drag their leg. So the arthritic marker you have checked from the plasma of the rat, you find there are a lot of increase of the arthritic and different interleukin level. So that model has been prepared, and then we challenge with the gold nanoparticles again that can effectively reduces the arthritis and all the markers. These are the characteristics of the nanoparticle. We have measured different markers of the arthritis, that is hydroxyproline, glucosamine, interleukin 1 beta, TNF alpha, and all these markers are significantly promising, shows us a significantly promising result against this gold nanoparticle tagged venoms. Then second paper is anti-leukemic potential of pegylated gold nanoparticle conjugated with the protein toxins. That is a lethal toxin from the cobra. And again, we have isolated, characterized different types of the particles. Then this is conjugated marker. This has been detailed characterized with the conventional method. And This is a resonance spectroscopy, FTIR, whatever the hydrodynamics analysis from the chemical analysis, and atomic force electron microscope analysis, atomic force microscope analysis, to characterize whether the conjugation is complete or not, whether the size of the gold nanoparticle is effective for the administration or not. These two are is a very important, by which one can easily ascertain with this sort of experiment. Then you again go for the IC50 dose of the gold nanoparticles. It effectively neutralizes the gold nanoparticles, and that gold nanoparticles, again, it is a fluorescence microscope result also shows that in two leucomic cell line, these are the leucomic cell line U937 and K562, these are the leucomic cell line, they effectively neutralize the leucomic cell line also. And this is a letter to editor. This is almost recent 2021. That is cobra venom neutralization by the gold nanoparticle. It is again that compound 2,4 2 4 methoxybenzoic acid. In very true sense, we are very keen to show that how this chemical compound is show the property equivalent to the antiserum. So our assumption is that whether there any antibody like molecule or antibody resemblance like molecules are present in that chemical pictures. Still, that work is continued by the student of Dr. Gomes. So they are trying to prove, yes, there are some chemical compound present in this chemical 2-hydroxy. Uh, uh, 2,4 2 hydroxy 4 methoxybenzoic acid, whether it is related with an antibody structure or antiserum structure present or Indian market, which is available in the Indian market. So we have compared the lethality, right, and also the stress. As because the different stress marker is already known, that is SOD, alkaline phosphatase, catalase, lot of molecule is there. So LPO, catalase, SOD, GS, glutathione. So all these markers have been tested. We show that yes, this nanoparticle also reduces the stress molecule. That means the protection is 100%. At present, we are now working the whether that chemical is equivalent to the antiserum molecule, because the antiserum molecule has a definite structure, is a y-shaped structure, definite region is there, FAB, FC, what is definite reason is there, and that region, the amino acid sequence is same with this 2-hydroxy 4 methoxybenzoic acid. And by the structural analysis, a structural chemist can only show or only prove the regions of present in the antibody is very much similar with the chemical moiety and the structure which is present with the 2-hydroxy and 4-methoxybenzoic acids. This is another work, that is osteoarthritis in experimental animal by the nanogold conjugated with the Naja cautia cytotoxins. And Naja cautia is a very potent. We are focusing the Naja cautia as because I have already mentioned that in India, the antiserum is not available against Naja cautia. It is available against Naja, big four. But what happens to Naja cautia, the Keute? So the bite of Keute is a very common also, but there is no protection system. So we suggest if there is any nanogold particle is there, so there is a chance of the protections against the bite of this animal. So against the osteoarthritis data, gold nanoparticle arthritis model is a good model to analyze the pathophysiological alterations. The upper table shows it is a control, another is arthritis, another is indomethacin, which is a normal standard drug. If you inject the mice can develop arthritis. This is a simple Naja cautia cytotoxin venom, purified fraction. And the last one is the Naja cautia cytotoxin venom plus gold nanoparticle. You find from the table that the gold nanoparticle significantly reduced the different ankle swelling, ankle diameters. So that can be easily measured. The different markers are also there for this measurement. It can significantly protect. This is also the different data, that is IL beta 1, MMP matrix metalloprotease, vascular endothelial growth factor, TNF alpha, interleukin 17, all these marker, these are the very promising marker for this arthritic disease. So we have measured thoroughly, and each time majority of the markers show the significant decrease if it is tagged with the cytotoxin and the gold nanoparticles. Then again, it is an expression of the tumor necrosis. This is TNF alpha, 1L beta, and the beta actin. This is scanning electron microscope of the knee joint of the articular surface of the collagenase. Now, look here, very food, the collagenase. Here, every time we found in the collagenase, similar result till now I have observed that yes, that is the collagen, that is something peculiar orientation in the snake skin. So that is the result why snake is not being infected with any bacteria, and that is the why this collagen plays a very important role in the osteoarthritic model in albino rat, and this gold nanoparticle can reduce this osteoarthritic level. That's proved in the scanning electron microscope. This is histological observation of the collagenase induced osteoarthritic male albino mice. This is SAM control. Look for the B, that is a gold nanoparticle, and the Naja cautia venom, and synovial membrane fluid. So bone staining is a very tough. So histology of the bone is not so easy. So the Dr. Gomes developed an indigenous method to cut the bone of the rat, and that histology can be takes place. And again, from histologically, we are also confirmed that nanoparticle is very effective to reduce all the histological alteration induced by the cytotoxic venom of Naja cautia. Next is the neutralization Naja-Cautia venom induced toxicity and the stress response by the Vitex negundi. There is another plant, right? Vitex negundi gold nanoparticle in experimental animal model. So another plant has been effectively checked. The same method, the characterization of the nanoparticle through FTI, to dynamic scattering and measuring the zeta potential, which is a very important zeta potential, and also the transmission electron microscope. And here there are the different markers for the Swiss albino mice. Urinary parameter have been collected, serum and urinary parameter, that is serum LDH, serum cytokine, serum GPH, serum GOT, that is glutamate oxalotransaminase, pyruvate transaminase, serum gamma globulin, serum alkaline phosphatase, acid phosphatase, urea, creatinine, calcium, and phosphorus. So all these are the group 1, group 2, group 3 mice, and group 3 is perhaps anti-venom treatment, normal conventional treatment, anti-serum, and group 6 is a nanoparticle, and there is Vitex negundi. So group 6 data shows a significant alterations of all these markers, and one from which concluded there is a reduction of the stress also. This pro-inflammatory cytokine, again it is measured, interleukin 1 beta, TNF alpha, 1 interleukin 10, all these markers have been similarly analyzed. That also shows the promising result against this venom induced toxicity. This is a histological observations, that is Vitus negundi and gold nanoparticle on the NKV induced liver damage. You saw the liver damage. A is a control, B is a only venom treatment, C is a anti-serum treatment, and D is a gold nanoparticle induced treatment. D is almost normal. If you tagged with the treatment of this Vitus negundi, gold nanoparticle causes partial recovery in the liver histology. Here it is a induction of the kidney. So this again, this partial recovery of the Bowman's capsule and all the damaged portions of the kidney. So this after this work, we have then in a position to formulate what are the therapeutically active nanoparticles conjugated venom toxins. It is from the bee venom. It is therapeutic role is acupuncture. Bombesin peptide, it is nothing but a yellow bee. That is a nanogold. It is anti-arthritic. Bufalin, it is from the bufo. Then it is a chlorotoxins. It is anti-cancer against glioma, that is a brain tumor. Melittin, again it is a bee. Polyethylenamine coated hexagonal phase, that is diagnosis and anti-cancer. Naja oxiana venom, it is adjuvant. Naja cautia, that our work, Naja gold, unpublished. Now it is a published. and it is an anti-cancer. Then potential venom toxin for future nanoparticle conjugations. So the source I have mentioned, the source, the potential toxin I have mentioned. We have isolated many compounds from the other animal too. That is one from the BM, that is Bufo We have isolated also from the shoal fish. It is a mural snake-headed fish. We have isolated Bhattacharya et al. So there are a lot of other particles we have isolated, and they have the terrible majority at the anti-cancer role. So the cell line used, they have been mentioned there, and the source have been also mentioned here. So DRCT, that means WRSLE venoms that have been mentioned. The several toxins have been isolated and used for nano tagging. So nano encapsulation bioactive compound from the animal venom. Again, I have make a table in details. So all the venoms have been mentioned here: the scorpion, the bees, and the snake, the scrotalus in African snake, and the use majority for this anti-cancer, and mainly these are the breast cancer cell line. So these are the table indicate the general applications of all the toxins and is used as a nanoparticles. This again the snake venom, the whole venom, where some of the whole venom is used, and some cases the PLO2 has been used. The site characterization they have given, the detailed characterization is very important before one can formulate the nanobody capsulations. The nanosystem used they have given, majority they have used the liposomes, some of the use the gold particles. So there are the many other methods, but recently the carbon nanotube has also been used. A lot of properties have been characterized. The biological activity is also being mentioned. Now, another paper which is also very recent, 2025, the neutralization of the Naja venom by green and chemically synthesized titanium oxide nanoparticle. This is look, the paper is from the Pakistan, right? So Dow University, but that opens a new dimension because the titanium dioxide use is new. Previously, no one can use the titanium dioxide. Number one, and the neutralization by the Naja, the species may be different, but this species is almost equivalent in India. India also Naja. I think the subspecies level may be different. So the snake venom affect millions of people, and the use of titanium dioxide nanoparticle by chemical and the green synthesis method utilizing potato peels and neem leaf extract. This is a very important because in the keyword you can find the potato peel and the neem leaf and the Naja venom. So that method uh, makes a very curious attention to all scientists because these are almost the green particles, and by using the potato peel and the neem leaf they use this nanoparticle, and they characterized, and the synthesis particle were characterized by scanning electron microscope, electron dispersive X-ray spectroscopy, FTIR, and the synthesis nanoparticle are spherical in shape and with a ranging from 41 to 64 nanometer, that is within the limit molecule. And this plant and this extract, this alkaloid plant extract, act as a reducing and capping agent, and these nanoparticles were evaluated for in vitro venom neutralization assay against Naja venom. The hemocompatibility assay showed that the titanium dioxide nanoparticle are non-toxic, indicating their safe use. The titanium dioxide nanoparticle were able to significantly inhibit venom-induced hemolysis of RBC and the blood anticoagulation effect. And this study suggests this titanium dioxide nanoparticle could be effective in reducing toxicity of the Naja venom, providing a practical and cost-effective therapeutic option to be used as an alternative therapy for the snake envenomation. So that is a very important highlights made by the group of scientists from Pakistan, and this journal is very high impact factor nano express. So there is a possibility of new tagging particle absolutely prepared from the green material, from the plant material. So another paper is that unlocking nature's arsenal nanotechnology for targeting delivery of the venom toxin in cancer therapy. So the liposome, different aspects. Again, it is an also very not recent. It is 2024. And this is the last paper what I have mentioned, unveiling the nanotoxicology of the snake venom through functional and biochemical characterization of extracellular vesicle. I have already mentioned in my first slide there is a possibility of this extracellular vehicle. Look this slide. I have already mentioned this is a latest therapeutic angle, the nanotoxicity use of extracellular vehicle. No one can till now focused use of the extracellular vesicle can be used as a just like a liposome. These vesicles are already present inside the cell, so that act as a natural nanocarrier for the toxins. So this EV can amplify the venom toxicity by interfering with physiological process like blood clotting. So this is the last paper, very recent. They have identified that these extracellular vehicles can be used for nanotoxicology, the functional biochemical characterization of extracellular vesicle from the naja and WRS, right? So this is a very unique and the first paper, and they can also, if you go through the abstract, you can easily find they can also reduces the venom induced hemolysis and all the reduces all the different enzymatic activity. So this is my last paper. Thank you very much for that. I must obliged for my mentor, Professor Gomes, all the research fellow who carried out this work, the funding agency, the principal, Maulana Azad College, and our collaborator, IICB, Calcutta University, and Bosch Institute. Thank you.
Thank
Thank
you,
you, sir.
sir, for your insightful and thought-provoking lecture. Now the session is open for the question answer. If you have any question, yes, I can see two questions in the chat box. So I am reading the question. First one is, sir, from Mithali Halamkar. Is the antivenom specific to the particular snake species?
No, antivenom is specific because in India, the antiserum which is available made by the Serum Institute of Pune, they are made against the big four snakes. The big four means it is one is cobra, that is naja, another is viper, that is W. russeli, another is a crate, that is Bungiurus cerulus, and another is saw-scaled viper, that is Ichthys carinatus. The antiserum which has been prepared against is a cocktail antiserum prepared against these all four venom. No specific single antiserum is not available in India. Is a
Okay,
polyvalent antiserum.
okay, thank you, sir. Hopefully, the Mithali Halamkar has cleared his her question. Now, the second one is from Manoshi Mondol. Sir, Barger's cerulus is
Bungiurus cerulus.
frequently seen particularly in May to August. As we know, its venom is neurotoxin. My question is that is there any specific antivenom available against its bite which can be stored in primary health center as from our college nearest hospital is Canning where AVS is available, which takes at least 1 hour 30 minutes, or any alternative way which we can apply for any emergency situation?
Yes, that's a very good question. So we have often faced that questions because usually bite occurs in the deep rural side of the Sundarban where there is no electricity, and the bite occurs during the night. So immediately after bite, so the patient is transferred to the rural health center, and the rural health center, if the antiserum is available, then it is fine. But usually the people come to the good hospital like Canning Hospital. But in the meantime, what the scientists suggested, administration or oral feeding of that plant extract, Hemidismus indicus or Vitis negundi, etc., etc. But till now, the antiserum treatment is the final option. And in India, only the Bungiurus cerulus vaccine is there because all the four, the Bungiurus cerulus is there. But if the bite caused by the Bungiurus fasciatus, then it is difficult to survive. No antidote is till date available.
So another question I have seen in the chat box, that is from Onnesha Choudhury. Beta cetosterol along with stigmasterol shows significant anti-venom activity by neutralizing various toxic effects of viper and cobra venoms. Can you please let me know which enzymatic reactions or cellular pathways are usually targeted by beta cetosterol or stigmasterol?
Yes, it's a good question, but we are not in a position to explain that, as I have mentioned, because this to know this detailed chemical function, how this chemical is synthesized and neutralized, we need a strong chemistry people. But from our laboratory, it is not being focused out. It is still open to for any work. I am not in a position to explain right now.
Okay, sir. Another there are too many questions in the chat box. Another one is from Ajoy Kumar Dash. Sir, can we procure snake venom from some suppliers?
Yes, if you have a proper license, otherwise they will not give you. Proper license, I means you have a proper research project from the government of India. This is number one. You have a proper permission from the forest department. Only then you can procure the snake venom. Otherwise, being a common man, it's very difficult to procure snake venom.
Okay, sir. Thank you, sir.
So another one from Rakhi Chakraborty. Is there any report of specific phytochemicals with anti-snake venom activity isolated from Vitex nigundu?
Yes, it's question, but as I mentioned, within 2 or 3 years, you can expect the nanoparticle tagged with the plant extract are available in the Indian market as a nano capsule or liposome tagged the plant extract nanobots instead of the antiserum treatment. That can be available, but not right at that moment. It is not. Within 3 to 4 years, it will come into the market.
Another one from Shoumabho Chatterjee. Is there any therapeutic approach to treat sepsis using venom nanoparticle?
Yes, there is a possibility to treat the sepsis induced by the venom nanoparticle. But to be very honest, we have not checked any antisepsis functions of the venom. But it was reported that the sepsis can occur through the snake bite. The gangrene is there. So, but we have not checked this antisepsis model. But the possibility is there.
Another one from Orunabhu Mukherjee. Suppose someone is bitten rarely by banded rat. Available antivenom can save him or her.
No, the conventional anti-serum what is available in Indian market is unable to neutralize this banded crate venom, right? So in that case, I advised you bring this that person as quickly as possible to a good hospital and charged with the available anti-serum along with other drug that depends upon the symptomatic symptom of the patient. Almost symptomatic treatment is there along with the conventional anti-serum treatment.
Okay. And the last one till I am seeing that from Nobonita Ghosh. Sir, is it possible to vaccinate against any snake venom?
No, after bite, there is a possible of the antiserum treatment. Previously, you cannot take any vaccine because it causes the terrible serum sickness.
Okay. Okay, Shoumabho Chatterjee has raised a hand. Yes, sir, you can uh, ask the question to the speaker.
Sir, I have another query. That is, in previous lecture you told about the nanobots, and here you are telling about the nanoparticles, antivenom preparation with the nanoparticles. In the previous lecture, I asked a question about the blood-brain barrier, blood-testis barrier removal. So, these venom nanoparticle encapsulated venoms are effective in crossing barriers and for such treatments, for such critical treatments.
Yes, it is a good question because almost all snake have a cytotoxic property and membrane toxic property. So obviously that can damage or rupture the blood brain barrier, right? Is inevitable. It can acts on the blood brain barrier. So using this nanoparticle to protect the blood brain barrier, that I have not we our lab have not worked, but till now I have not noticed any paper which is concerned about this the blood brain barrier rupture protections. But in near future, I think that study should be done, because that is a very important criteria for any snake venom. And as I mentioned, that a new approach has been already started in Indian science by the Indian Institute of Science Bangalore group of scientists. The tiny nano robots or tiny nanoparticle or nano capsule has been in the preparation, is on the way, substituting the conventional anti-serum treatment. Probably that nanobot or nanorobots may counteract this nano blood-brain barrier rupture concept.
Thank you, sir. Thank you very much.
So another question I have seen in the chat box from Mithali Halankar, that as snake venom has therapeutic activities, can nanoparticle based approaches be used to deliver venom toxins that have therapeutic effects? Are there any studies done on that?
Yes, yes, that several studies have been that, but the conjugation of the nanoparticle and the snake venom and the therapeutics is very recent, and a very restricted group of work has been work carried out throughout the world. So I think within a few years that area has been gained much attention, and your proposal or your thoughts are in a full-fledged function in that time.
Okay, sir. And
Sir, I have a question. May I ask? Sir, this is for
Yes,
Maraha. Yes.
yes.
Sir, you worked with the gold nanoparticles. So, as we know, with the nanoparticles, size is an important criteria for its functional aspect. So, has there been any changes with the size parameters by changing the size of the
Yes,
gold
yes. If
nanoparticles?
this, yes, absolutely good question. If the size is not within the permitted limit, then it will not show any effective measure, right? It will not goes for the counteractive measure. It will not show the therapeutic sense in terms of the neutralization, in terms of MLD. So before one can proceed for the nanoparticle, the characterization is a very, very important factors, the size, because the nano means the size is very much important, no? And all these size can be characterized by all the different angle, AFM, DEXA scan, FTIR, and it's rather involvement of too many sophisticated unit, and also the chemistry people. That point I am repeatedly hammering, that along with the zoological background, it's difficult to carry out the nanoparticle research. You need a strong chemistry background. Till just now or just today, I am working with an XRD, right? The XRD of the skin of this snake. But I am not a specialist of XRD. We use the term XRD in case of the DNA double helix made by the franking vehicle. That's all. But how the XRD analysis can take place, what are the peculiar combinations of the collagen structure, that is absolutely carried out by the chemistry people. So the size is very, very, very important.
Okay, thank you, sir.
Okay.
thank you, sir. Are any questions from the participants? Okay. So thank you, sir,
Thank
for
you.
your deep expertise in nanomaterials and venom toxinology, cell biology, has once again given us a clear understanding of current progress as well as the challenges that still need attention. Yes, Dr. Namrata Roy has some queries, I think. Okay, thank you. For integrating
And
green nanotechnology.
I have just one thing to ask. In my first lecture, some participant asked
Yeah,
me a
yeah.
good
Dr.
question.
Orunima Dotto has some queries regarding your speech. So, is Orunima ma'am here?
Yes, sir. I had asked you that about the fangs, the fang marks that I asked you.
হ্যাঁ ফ্যাং মার্কস,
And
গরিব গ্রামের গরিব লোকরা তো ফ্যাং মার্কস জানে না, বুঝতে পারবে না, কথাই বলতে পারে না, সই করতে পারে না। ওর ফ্যাং মার্ক জানবে
That
কি করে?
that I have understood, sir.
হ্যাঁ, না আমি বলছিলাম আগের লেকচারে একজন আমাকে জিজ্ঞাসা করেছিল ন্যানো পার্টিকেল ইন ফ্লোরিকালচার অ্যান্ড এগ্রিকালচার। আমি ওই
that,
উত্তরটা দিতে পারিনি, কেউ একজন জিজ্ঞাসা করেছিল।
that might be
But
from somebody from botany, sir.
হ্যাঁ, বটানি, রাইট। কিন্তু উনি
Botany.
যদি থাকেন তার উদ্দেশ্যে আমি বলছি, আমি যেহেতু কোশ্চেনটা খুব ভালো ছিল এবং ন্যাচারালি আমারও একটা ইন্টারেস্ট হয়েছিল তখনই যে ফ্লরিকালচারে যদি ন্যানো সায়েন্স ব্যবহার করা যায় তাহলে তো ফুলের খুব ক্রাইসিস, বিশেষ করে বর্ষাকালে ফুল হয় না। তো আমি একটু সার্চ করেছিলাম, পড়াশোনা করেছিলাম ফর দ্যাট পার্টিসিপেন্ট। ইউ মে সার্চ ফ্রম দ্য গুগল। ইয়েস, দেয়ার আর লট অফ ওয়ার্ক হ্যাজ বিন অলরেডি ডান with the floriculture and the nanoscience, right? Lot of work. In agriculture, there are extensively use of the nanoparticle for the productions of the cereals and other vegetables also in the floriculture. So requesting that participant to go through the literature through PubMed or NCBI or Google search or ResearchGate, you can easily find lot of particle that can solve your query.
Sir, one thing, this is Orunima. One thing I would like to ask that I have read about nanoparticle toxicity itself. So, if we are using nanoparticles to combat the venomous conditions or the effect of venom in body, won't there be any toxicity caused by the nanoparticles?
Yes, that's a good question. So the toxicity is depending upon the size, right? So if the size is too much beyond limit, that can causes the toxicity, because gold itself is toxic.
Yes.
So if you enlarge or if you cross the limit, then that gold causes the toxicity. Instead of neutralizing the toxicity offered by the gold itself.
Sir, in one of our lectures, if I'm not taking much time, I just want to ask this question, madam.
Actually, it's already 12 and the next speaker is on the way. So
Take 2 minutes, ma'am.
Okay.
Sir, about this gold nanoparticle conjugate, in one of our lectures in the series, we have learnt about plant viral coats can be used in place of animal viral coats to deliver our nanoparticle of choice. So, can it be replaced by these plant viruses?
Yes, that can be possible, but no what till now. That can be possible.
That work hasn't been done, or
No. This is new area, no, absolutely new area.
So we can combat that nanoparticle toxicity itself, because plant viral coats won't have any effect on synergistic effect.
That's very difficult to say. That's very difficult to say.
Okay. Thank you, sir.
Thank you.
Thank you.
Okay, thank you.
Thank you, sir. Thank you once again for this wonderful session and answering all the questions patiently. Thank you so much.
Okay, thank you.
All the participants, the next session will be start very shortly. Please stay with us.
So, good afternoon, everyone. As part of the Malaviya Mission Teacher Training Program, we warmly welcome you all to the online refresher course on Bioscience, Bioengineering and Nanotechnology from 2nd December to 15 December 2025. We are here on the 9th day, and we will be starting with the 26th lecture, to be delivered by Professor Sanjukta Patro from Department of Biosciences, Bioengineering and Bioengineering IIT Guwahati. Ma'am has already joined us. Even though she needs no formal introduction, but basically to say a few words about her, Professor Sanjukta Patro completed her PhD from Central Food Technological Research Institute, Mysuru. She has been working as professor at Department of Biosciences and Bioengineering at IIT Guwahati. She has headed School of Agro and Rural Technology at IIT Guwahati from 2020 to 2023. Currently, she is a faculty at the Department of Medical Sciences and Technology at IIT Madras. Dr. Patro has been actively engaged in research and developmental activities in the areas of enzymes and microbial technology, with a major focus on enzymatic and microbial applications in different spectrums of diagnosis and therapeutics, with focus on translational research. She has contributed in diagnosis of malaria, tuberculosis, systematic fungal infection, to name a few. She has, to her credit, 7 granted patents, 71 research publications in peer reviewed journals, 30 book chapters, 70 conference proceedings, and the list goes on. So far, she has guided, and under her guidance, many students have got their PhD degrees awarded, and many are pursuing their PhD degrees. She has also been mentoring master's students. Her research team has 3 postdocs under SERP, National Postdoctoral Fellowship Scheme, 2 international exchange students under DBT TWOS scheme, and DST RTF DSC scheme, 1 DST Young Scientist. She has completed 15 sponsored projects, while 3 of her projects are ongoing. She is a life member of Indian Association for Cancer Research, Association for Microbiologists of India, Society of Biological Chemists, Association of Food Scientists and Technologists, Biotechnological Research Society of India, and Biosensor Society of India. She is currently serving as editorial board member of the International Scientific Journal, Scientific Reports, and various other journals of her research domain. She has recently edited 3 books which got published by Springer Nature. These are Nanobiosensors for Point of Care Medical Diagnostics, Enzyme Based Biosensors, Recent Advances and Applications in Healthcare, and Technologies for Rural Development. Her wide, holistic level of research and development is quite understandable by the research background she is having. We are extremely blessed to have a lecture with us today as part of this online refresher course. Today, she would be speaking on sustainable solutions for monitoring of antibiotics and assessment of water health. Over to you, madam.
Thank you, Titasha, for this nice introduction. I'm extremely thankful to you and the organizers for inviting me for delivering this lecture. Before I start with, I'll definitely take over in a few seconds. But before you start with, may I know, a little bit about the background of the audience, so that I orient my talk in that direction, like whether they are from biology background or from mixed background, so that I maintain my depth of my talk in that way.
So most of the participants here are from bioscience background, as far as I think. If others like are from any specific specializations, please feel free to unmute yourself and say your specialization to ma'am.
Madam, I'm from a chemistry background. I'm Dr. Namrata Rai.
Okay, great.
Some are from zoology also.
Okay. So mostly different areas of biology, chemistry, and other things, right?
Mostly, ma'am.
Okay, good to know.
And just a second, ma'am, before you start on, I would say that please put in your questions in the chat box, or else we can have an open discussion once when ma'am finishes with her lecture.
Yeah, I'll be happy to take up questions from everyone. Just share my slides, 1 second.
Definitely, ma'am.
Can you see the slides?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Visible.
So is it in slideshow mode now?
Yes, ma'am. It's in slideshow mode now.
Thank you so much. So good afternoon to one and all. So today I'll be speaking on sustainable solutions for monitoring and remediation of antibiotics and assessment of water health. Before I start with this, I'll just give a very brief background of my academics, wherein I am basically a microbiologist who took up biochemical research, biochemistry research, and finally I am into. I have developed my group basically works on enzyme and microbial technologies and diagnosis and therapeutics. And in this domain, we also work in monitoring of water health as well as their remediation using enzyme and microbial technologies. Now, water is a very essential part of all of us our life, and we need water for various applications. And based on that, the water quality requirement also varies. Now, if you talk about water pollution, that limits the accessibility of water in our life. And this leads to what we are coming up with something called water stress. Water stress is basically a term used all over, and this stress is percolates different regions of the world in its own way. If you talk about India, India has its own water stress map, which you see here, where we see the number of people who are affected in terms of the number of people who are affected in terms like 600 million people face high to extreme water stress. 75% households do not have drinking water. 84% rural households do not have piped water access. 70% of our water is contaminated. And why all this? So we have water but not accessible water, so that is the point. So when we talk about that, we basically then come down to water pollution. And when we talk about water pollution, besides everything which we have been hearing since we started school education, maybe we have been talking about water pollution, we talk about different things present in water, starting from heavy metals to pathogens to many other things. But the recent addition, one of the most recent additions, had been antibiotics. And the addition of antibiotics to water causing being a contributor to water pollution is mostly because of the overuse of antibiotics. And we are not being wise enough to use antibiotics. Otherwise, antibiotics are life saviors for us, but when it enters from the self, it goes to the environment. There is what it does, the damage to all of us. And because of which we are also facing something called antimicrobial resistance present both in environment as well as antimicrobial resistant in terms of the diseases which are picking up antimicrobial resistance, and they are stopping to, and they have stopped, they are now turning they are turning away from responding to antibiotics for which they are made, and hence the disease percolation keeps on increasing. So with this background of antibiotics, when it enters the environment, I start with a talk wherein we see that antibiotics are otherwise required for various forms in our life, but they, once they are released into the, once we are not disposing it properly, and it goes into the environment, and where we see that because of, and currently we do not have water treatment systems, or what we call water treatment systems, which can take care of this antibiotic remediation. And when the water comes back after treatment to us, they are actually not free of antibiotic and again re-entering us. Now the rise of resistance in antibiotics, if we see, it has been there since what we see, that penicillin resistance was observed way back when it was discovered even, but it was all contented to a very small zone, and slowly, if we see here, by 2010, even the best of antibiotics have become resistant. We have developed resistant against the best of antibiotics also. Now this is what we see is the fate of antibiotic, how it comes. So you have the natural and synthetic production of antibiotics. So overused and mismanaged, they get into the wastewater treatment plants, where they get into the plants and to the soil and the plants because of the water which is going there, and some of them get degraded and some of them not, and then we come across what we call antibiotic resistant microbes, and again they are back to the square one, where we are facing, where humans face the resistant microbes, or that there are threat to humans. Now antibiotic use and abuse, if antibiotic abuse, if you talk about the consequences, so it increases it leads to increased human mobility, increased human mortality, reduced efficacy of related antibiotics used in humans, increased human healthcare cost because of this, increased potential for carriage and dissemination, facilitated emergence of resistance human pathogens. So these are some of the consequences of this. Now, the commonly consumed beta-lactam, if you talk about one of the examples, commonly prescribed fluoroquinolines, macrolides. So these are, all of them are leading to antibiotic resistance and the consequences which we see. Now, if antibiotic is present in our day-to-day drinking water, how do we know it? Or even when it is being released to the water bodies and finally the rivers and all other places, how do we know it? So it is important, before we start to remediate antibiotics so that they do not reach us, it is important for us to know how much antibiotic is present in water. So that is what brings us here. So antibiotic sources, if you talk about, the sources could be many, like right from agriculture, aquaculture, poultry, water and animal usage, hospitals. So all this lead to release of antibiotics into sources like the water bodies. And but how do we monitor them? So that is what we are going to talk about, because if you talk about antibiotic pollution and monitoring, the conventional methods which are still in use are cumbersome. If you see here, some of them are HPLC based methods. Many of them are HPLC based methods, LCMS based methods, and there are very few methods which are quick to use, and it is not it doesn't take trained manpower to handle, and also in terms of the time taken for monitoring. So with this, the points to note is that no commercial application for the existing methods, no regulation on the release of antibiotics in the water streams, little to no regulation on the presence of antibiotic residues in food commodities. And so how do we move forward? Before we move forward to remediation of where they are present, they are generally present in both abiotic and abiotic forms, aerobic treatments, anaerobic treatments. And so these are the things which are taken up for remediation. Physical and chemical methods could be there. So these are the remediation methods. But before we reach the remediation methods, we are going to talk about today about development of a very simple to use antibiotic monitoring method. And the three antibiotics which we have listed out here is ciprofloxacin, amoxicillin, and tetracycline. And why we have listed these three only? Because we carried out an initial study uh, wherein we listed the most abundant antibiotics which are in India, because we try to see the scenario in India. So we listed out almost 13 antibiotics, and out of them ciprofloxacin, amoxicillin, and tetracycline were found to be among the highest ones which are being used. So we have chosen these three antibiotics, how we can come up with monitoring for this. And for this, the sensing platform which we have chosen is an MIP based sensing platform. So MIP is basically molecularly imprinted polymers. Now, what are they? Many of you might have heard about it, especially people who are from chemistry and who work on sensors. So molecular imprinted polymers are basically a polymer cage. You can see here, you can say a polymer cage which is created against a particular target. So herein, it is antibiotic, and all three antibiotics which have chosen will have different MIPs. So each molecule, each small molecule which we have chosen will have its corresponding MIP, and how is it created and what is it basically. So if we see here, so if this is the template molecule, suppose one of the antibiotics, so what we do is against that we have a we choose a monomer which will go for formation of a polymer again around this. So the polymer will be formed. So if you see here, so this is the polymer which is formed against the target. So this is the target. It can be any of the, suppose ciprofloxacin. Now after the polymerization, what we do is we remove out this target. So what is left? So this is the template removal or the target removal. So after it is removed, what is left is basically a cage, which is exactly the same imprint of the target molecule. Herein, suppose we have chosen ciprofloxacin, so this cage is of cipro here, and any other molecule will not fit here. It's only cipro which will fit. So this tells about the specificity of the cage or the molecular imprint which has been created against any target molecule. So it's highly specific. That's what we are trying to say. And because it is highly specific and gives a one-to-one reaction, hence they can be used for monitoring of a one-to-one target molecule what we choose. So how do we create that? We will talk about a little bit about that. So for each of these, what we had done is, there are many monomers which you can try with against any molecule. So the monomers which we have tried, if you see some of the common monomers herein are acrylic acid, acrylamide, methyl acrylamide, pyrols. So we tried styrene. So we tried all these monomers against each of these antibiotics. So we try to come up with what will be the best monomer. So we did some computational studies against this, and to see that what gives the what will be the best monomer for creating the polymer for the MIP. So if you see here, acrylamide was chosen for ciprofloxacin. For tetracycline, it was methyl acrylamide, and for amoxicillin also it was acrylamide. So based on that, we choose based on the computational studies, we choose three different monomers against which we were going to prepare the molecularly imprinted polymers, which will act as these monomers will form the polymers. So if you see here, this is ciprofloxacin against which, and this is actually the computational studies which is saying that why this is being chosen. It is because of the bond length and other things which will give us a very stable polymer, and so that the cage is created very stably, and it will also give it a higher shelf life so that it can be used many times. So this is for amoxicillin, and this is for tetracycline. Now, if you see here, now the point is we have made a molecular imprinted polymer where we know that that the molecule of interest, if it is present in wastewater, so ciprofloxacin is present in wastewater. If it is allowed to interact against the MIP, it will go and interact. So the antibiotic will get trapped in the MIP. Okay. But the next question is, after getting trapped, how do we get it out so that we can use it for further monitoring? So we want the antibiotic from the MIP, and for the MIP to be retrieved from the wastewater so that it can be used. How do we do that? So for this, what we did is we synthesized magnetic MIPs. So they were not just MIPs, they were magnetic in nature, and because of which they could be separated out after we allow it to interact in wastewater. So these MIPs were magnetized, and when we allow it to interact in wastewater, the antibiotic is going to come and bind in the MIPs will come and bind in as their templates, and after that, because they are magnetized, we can separate out these MIPs because of their magnetic nature which we embedded in them. Now, these are some of the characterization methods which we carried out during the synthesis of the magnetic MIPs, wherein you see that all of these have been characterized using TEM and then EDX, and which tells about basically the features which they have and the structural features which they have when we talk about the MIPs of the different antibiotics which we were working with. Now, this is the characterization of synthesized magnetic MIPs using FTIR XRD. So this data shows basically the characterization using FTIR XRD and DSC, where we talk about the how the polymerization has taken place, which confirms not just the polymerization, but also the monomers which are in it. Now, after that, so basically now we have magnetized MIPs for each of these chosen antibiotics. So after that, what did we do? We tried to see their performance, and we try to characterize them further, like at what pH, because wastewater will have will have a varying pH. We don't have a control over the pH of wastewater, right? So for that, we try to characterize it for its pH, like at what pH it will work, the adsorbent amount which it will be able to absorb, the incubation time. Just one second. Just 1 second. Hello. Yeah. Sorry. Oh yeah. Actually, I'm into a talk. Can I call you back? Can I call you back? I'm into a talk. Thank you. Extremely sorry. Yeah, so what are the features which we optimized, like the pH, at what pH these MIPs will work, and at what pH they will fall apart, the adsorbent amount, how much they can absorb. So basically here the dosage of antibiotics which can be worked with, the incubation time, or the time to which we should allow for the antibiotic present in wastewater to interact with the magnetic MIP. So these were some of the things which we optimized. And so this is for amoxicillin. This data is for amoxicillin, as we see here. So if you see here, if you talk about the incubation time, at around 30 minutes of incubation time, so maximum of the things were we could got absorbed onto them. And if you talk about the pH, if you see here, so at this pH it was best at around 7, so that takes care of most of the things. And this was for tetracycline, if you see here. So all of them were behaving at around 30 minutes, most of them responded. However, like if you see, like tetracycline could work at almost all pH. It responded to all pH. Now we also checked, so it was important for us because we developed it against a particular antibiotic, and that is why MIPs are that's the characteristic features of an MIP, to be very specific. But still we wanted to be sure, like whether they are, how selective are they, how selective is the adsorption and adsorption. So for what we did was, against the MIP which we had developed, we also like, even if it was for Cipro, we also tried to see like whether similar antibiotics will be picked up by them. And if you see here, if you see this particular graph, so we see that Cipro has been picked to almost more than 90%, but the other antibiotics which has been picked is really very, very less. So similarly for the amoxicillin, if you see here, and for this is for tetracycline. So there is no cross reactivity, or negligible cross reactivity, I should say. And so this we could see from this particular data, like how it is being highly selective. Now, once it is the absorption has happened, the next thing was about how do we MIP has captured the antibiotics from wastewater. So the next thing is, how do we find out how much is the antibiotic? So that is based on what we did next was, we try to dissolve the antibiotic from the MIP, or rather take out the antibiotic and then go for a simple spectrophotometric study. So for that, what we so for the desorption, if you see, so after the incubation of 30 minutes, desorption was carried out with methanol. So this is actually a very simple thing which we have designed. So this is basically, these are filtration units. These are small filtration units which are very much equivalent to Eppendorf's. So if you see here, so this is this has a filter kind of thing. So in this, we allow 5 mg of magnetic MIP to interact with wastewater, whatever you want to check for antibiotics. Okay. And after 30 minutes of incubation, which we had already optimized, we allow it to the water to flow out by opening this by opening this. So when the water flows out, what is retained back is only the magnetic MIPs. So these magnetic MIPs, which have captured the amount of antibiotic which was present in the wastewater with which it could interact, now this antibiotics can be dissolved. How do we dissolve it? We can just allow it to interact with methanol for 5 minutes, and the antibiotics will now come out from the magnetic MIPs. Now, once these come out, so we can just go for a spectrophotometric reading. So as we see here, so yeah, we go for a spectrophotometric reading, and so this is what to what extent we can. So this is take UV absorbance for the collected elute, and each of these antibiotics will have their own lambda max against which we can measure it. So here we see that for magnetic MIPs of ciprofloxacin, the linear range of detection was 100 to 500, and this is the R square value. So the lowest, the LOD or the limit of detection was as less as 0.03 microgram per ml, which is way below the amount of antibiotic which is generally present in water, and which is for water to be declared healthy, the amount is quite we have gone much below the lower limit of detection. We have taken down quite low, so that we can really assess everything, almost everything which is present. So this is for amoxicillin, and this is for tetracycline, and as we see that the limit of detection is quite low for all of these, and it takes only this simple way of. So what are the things which we have done? We have created an MIP against each of these antibiotics, we have magnetized them, and we have devised a small filtration unit which can act as the interaction point, and then it can be dissolved under simple spectrophotometric reading, which can be done by by anyone in a very simple way. So this was the entire things which we have developed. So this was all about the detection part which we are talking about, which we have developed. Now coming to the so we can monitor and say which water is healthy and which is not. But if something contains antibiotic and we need to remediate it, what all things can be done? So towards that, we came up with an advanced biooxidation process for sustainable remediation of antibiotics, wherein we try to use enzymes. And it was not just a particular enzyme, it was a combination of enzymes which does what we call the biooxidation, which we call the biooxidation system. So what does it do? It basically creates, so these group of enzymes create what we call hydrogen peroxide, and hydrogen peroxide can be broken down into, because they are not very stable, so they will break down into what we call hydroxy radicals, and hydroxy radicals are supposed to be very highly reactive in terms of their reactive oxygen species, and they can they're derivatives of oxygen that are more reactive than molecular oxygen itself, and hence they can help in breakdown of many things, including antibiotics, as we have seen here. So this is the standard reduction potential in aqueous solution, and we see here that hydrogen peroxide is at a very high occupies a very high position here. So in fact, it is more than molecular oxygen. So hydrogen peroxide, when breaks down into hydroxy radicals, it can be used for cleaning up of antibiotics as well, and this can be clubbed with Fenton reaction, which can make it even better. So the combination of enzymes which we have taken here is glucose oxidase. So we have taken three different enzymes to create the oxidation potential which can remediate the antibiotic. So the three are glucose oxidase, lactase, and manganese peroxidase. So what we have tried to go for is we have tried to go for a CLIA process where which tries to get aggregates of all these enzymes together, and when this interacts with antibiotics, we'll see how do they remediate it. So here again, we try to develop an integrated remediation system for degradation of antibiotics, where you see these are the three different enzymes where we have chosen, and we try to amine functionalize. So we have again these were amine functionalized and precipitated. So and then finally cross-linked to what we get it here. So we got magnetic nanoparticles where all these three enzymes were present in a combined form. And these, when allowed to interact with antibiotics, they degraded these antibiotics. And we see here, so we try to so these are these enzymes so what we also try to find out, are these enzymes interacting with the antibiotics as a enzyme substrate and giving an enzyme substrate degradation, or otherwise, or what we claimed was the advanced bioxidation process. So we see here that through isothermal calorimetric studies, we see here that these antibiotics were not the substrates for the enzymes, and they did not bind and give us the indication that they were binding in an enzyme substrate reaction. Rather, as per our claim, we saw that they were trying to give us the hydrogen peroxide, which was broken down, and gave us the hydroxy radicals, which did the remediation. So these are some of the data which wherein we cloned and expressed the one of the major enzymes, which is glucose oxidase. And so this was cloned and purified and characterized. So this recombinant glucose oxidase was actually used so that we could reduce the economy of the process as such. So and these are the different development of an integrated remediation system for the biodegradation of antibiotics. So wherein we took different all these three enzymes, how we can try to develop the CLIA of that, and we did that using the triphase partitioning process. And the functionalized enzymes, so these enzymes were also functionalized so that they can be used, because we were not looking for a one-time use, we were looking for a multiple time use, and hence they were also functionalized magnetically so that they can be retrieved and used again and again. So here we see the different ratios which help us, different ratio of these enzymes which help us to get the CLIA. And finally, we try to, these are the functionalized enzymes and showing the residual activity of the enzymes after each use. So this is the characterization of the magnetic CLIA which we did, which was using the SEM and TEM. So here we see that the enzymes have been we were able to immobilize the enzymes on all the three enzymes which we used, where we could immobilize them on the magnetic nanoparticles. So these are the further characterization process using EDX and FTIR. And we try to see the steady state kinetics of the free enzyme. We also try to see like how the free enzyme works, and whether the immobilized enzyme, and the difference between the free enzyme and the enzyme and the immobilized enzyme. So this was only glucose oxidase, manganese peroxidase, and lacase, and this is all the steady state kinetic analysis of the of the magnetic CLIA of all the three of the immobilized enzymes. And then we did the temperature stability of the free enzymes as well as the immobilized enzymes, which we did by CLIA. So this shows that they can work in a wider range, and when we talk of its utility in wastewater for remediation. So this is a time-dependent degradation analysis of the target antibiotics by free enzyme and by the magnetic CLIA. So if you see that it is degraded quite fast, if you see here, all of them are degraded quite fast. And after that, we tried to also see like in real water samples where we had wastewater, and in that we antibiotic was added, and we saw like how they could. So basically the parameters of wastewater will also have many different things which could be inhibitory in nature. So we saw that our CLIA could perform, our enzymatic CLIA could perform in wastewater also, and at the same time degrade the antibiotics. So this is the degradation percentage which you see here of amoxicillin, which is reaching almost 100. This is tetracycline and CI and ciprofloxacin acid. So this and these are the pH at which, and we took different waters. So we took river water, sewage water, and even groundwater. So and checked the performance of the CLIA which could degrade them. Now, after degradation of anything using any method, whether it is physical, chemical, or biological in nature, it is also important to see like if the antibiotic is being degraded by our method, are they producing any species which is more toxic than the antibiotics themselves? So for this, we carried out we carried out LCMS analysis of the degradation products, and we tried to identify them. And we'll see some toxicity studies further. Before that, I would like to say that the reusability of the magnetic clear was also checked, and we saw that we could use it for till 6 times, though the performance decreased. So this is the first time performance, second, and slow it kept on decreasing. So here we see that we could use it till 6 times, but of course the till third or fourth time it was really good, and after that it decreased a bit. Now we'll come back to the toxicity studies, where the species which was degraded when the antibiotic was degraded, whatever species we got, or the molecule which was degraded, the antibiotic molecules, were they more toxic, or how toxic were they? We tried to find out that. So this is growth inhibition assay and cell viability assay, where Bacillus subtilis, a strain of Bacillus subtilis, which is well known for carrying out for assessing toxicity of degraded products, were used. And we could see, because we could see that the degraded products were not more toxic than the antibiotics themselves. So this is a flow cytometric study, where we see that the plots of the where propydium iodide was used to check the permeability of propydium iodide, and see that and to see the invasion of the toxicity of the degraded products. Now, the second thing which we did was second toxicity study to confirm this. What we did was a bioluminescence inhibition test, which is by using another strain of bacteria, which is Alevio vibrio fischeri. So this gives luminescence, and it's a very sensitive assay to say that if the organism is if the product is more toxic, then the organism cannot grow, and it will show, and the luminescence will reflect the toxicity. So from both these studies, we could find that none of the antibiotic were degraded in a way they did not create any the further degraded products were not toxic, and hence could be the water becomes safer. Now that so this was so to summarize, this was development of an enzymatic process where 3 different enzymes were used, and which work via the biooxidation mode, advanced biooxidation process. And here we have generated a magnetic clear which can be retrieved for reusability, and this can target this can target all these 3 different antibiotics which we talked about. And the bioremediation by the enzymes, more than 90% of degradation in the first 4 hours. So the time, if you see, of degradation was also quite less, and the excellent reusability up to 6 cycles we could see. And the enhanced temperature and pH stability applicable on different, as we saw that it was in wastewater from river as well as in groundwater, and it could respond to all of them. It could perform in all of them rather. So with this, I would like to thank you, my entire team. So this was a PhD work which was carried out by Tarun, and a part of it by Satyam, who have submitted, and we are now currently into their postdocs. So these are my, almost 17 out of 25 students who have graduated with me, and some of the postdocs and my entire group. And I'm really thankful to all of them for contributing to the team. Thank you. I'm open to questions now.
Thank you so much, ma'am, for your illustrious lecture. Now the house is open for questions. Can we have, please, questions? I can see as of now there are no questions in the chat box. So I would request the participants that you can even ask something that you want to clarify. Ma'am is here to answer to all your queries. Okay, so you can unmute also and ask questions. Is there any question from the participants? I think there should be some questions after such an illustrious lecture. Ma'am, is there anything you want to further like point out or?
No, I would be happy to take up any questions if there are any in any of these things, like any of the basic things also, or whatever work we carried out. I have nothing to say from my side. I'll be happy to take up questions if any. Otherwise, I'm done.
So, ma'am, there is a question from Orijit Das who is asking, ma'am, can you tell us anything about antibiotics in soil?
Yeah, so good question. Antibiotics definitely, as we saw in one of the slides also, like through water it goes to the soil. That soil is definitely one of the matrix where antibiotics accumulate. And not just accumulate, they also do their own job in terms of, again, creating antibiotic resistant microbes also. So soil is definitely a matrix where antibiotics are there. And if we want to monitor like if antibiotics are there in soil or not, definitely what we need to do is you have to make that soil, from that soil you have to get it in soluble form. Basically you have to dissolve it in water, and then if you once you remove those soil particles by centrifugation, or just settle it down and get the clear water, you can use it for monitoring. So if it is with regards to monitoring, it can be done that way. Anything specific, Orijit, you want to know more than this?
You can unmute and even you can ask. Sir, you can unmute and ask.
No, ma'am. Thank you from my side.
Any other questions? We can take, we have some time, so we can take questions if you have any. Okay, so Durba Bhattacharya, madam, you can unmute and you can ask your question. Ma'am, there is a question from Durba Bhattacharya.
Please.
Madam, thank you for your very informative lecture. I have a question, is that when antibiotics enter into the environment, especially, there are many variables which come into play, because there is temperature and um, temperature, where it is going, whether it's in water or in soil. So much of variables are there. So as you are, and especially there are a lot of problems in disposal of antibiotics also. There are no, I think there are no specific rules or regulations for disposal of antibiotics. So do you think that there is a need of are that there should be some specific rules for disposal of antibiotics?
Yeah, Durba, very relevant question, I should say. You are right to start with. There is no policy where we are guided like this is the way you should throw away your antibiotic. Today, we have policies about even electronic wastes, and of course, the other common wastes are always there, which are it's a framed thing already is there. But for antibiotics, we do not have, and unfortunately, see, if when we talk of antibiotics, each of us are contributing in terms of the fact we don't know, and we don't actually, there is not a policy, so we throw it as individuals. We may be throw it even to our household dustbins. But the what I will say, when we see that even the pharma companies and the hospitals, they are actually, from their side also, there is no rule by from the government where they are also guided, because they are doing it in bulk. Of course, each one of us are also adding, but they are doing it in bulk. So there is basically no guideline which neither tells them how much to release, nor like how much they should take care or remediate before releasing them into the environment, whether it is water or soil. And in this context, actually, before coming up with this study, we checked even hospital wastewater. So their STPs also, we tried to get the water and check. And unfortunately, from their treated water as well as their wastewater contains lots of antibiotic. So to summarize, I will say definitely very you know, to start with, there should be guideline, and these guidelines has to vary from user to user. Like if it's an industry, how should they release their wastewater to the main water bodies, and so. And if it is hospitals, how should they treat their wastewater before they release. And if as individuals, where should we go and throw the antibiotics. So I think these things are really required to be put up by the government. And these kind of studies, I think many studies are coming up when we talk about antibiotics. So all of these studies put together will probably help in getting some guideline for the environmental load which is to be fixed up, I should say. Did I answer you, Durba?
Yes, thank you, madam, for the clarification. Thank you so much.
Okay, so we have a question, ma'am, from Oyun Das. Can the presence of antibiotics in drinking water be detected via any kit at home?
Yeah, so as of now, I will say, Oyan, that good question. And as of now, there are no kits which say detects antibiotics in water. And this quick method, what I talked about, we are trying to come up with one. Like if you see, this is a very simple thing, and if you can basically connect it with if not a spectrophotometer, because we just need an absorption in the UV range. So if you have a miniaturized version of that, that probably can help. But otherwise, there is no kit as such, to the best of my knowledge, which exists in the market, which can talk about drinking water things. But you can get it tested somewhere in labs or so.
Okay, ma'am. Thank you so much.
Okay, so any other questions? Water and water stress are things which are very close to us. So the presence of antibiotics in them also cause a lot of concern. So do we have other questions?
Madam, I have a question. Hello?
Yes, please proceed.
Okay, madam. Thank you for your such an informative and illuminative lecture. My question is that is there any lifestyle and community level actions can reduce the spread of this type of drug resistance bacteria or like that?
Yeah, definitely so. Yes, we do have a role to play. First of all, we should not, nowadays, I think we are too much panicked whenever we have something. Even if we have common cold, we just want to get rid of that common cold as soon as possible. All of us, we don't want to lose our time, and we want to get rid of it. We don't allow our body to fight against it because it takes its time, right? We may not have time, but then and we rush and we have antibiotics. So when we talk of antibiotic abuse, basically it is called, we should really think like what is the point at which we should start even think of taking antibiotic. So it starts from there. So even if you have to, so first of all, think before you do, we should think before we take antibiotic. Next, if we are taking antibiotic, are we completing the dosage which the doctor has prescribed, or we are, because after two or third day I start feeling good, so am I stopping the fourth and fifth day? And when I stop, we don't go and return back the antibiotics to the hospital or wherever we took it from, right? Because there are ways of disposal, also certain ways, like actually we should go and return back to the hospital. But I think none of us do that. What we do is we put it into the dustbin, right? Maybe may not be the fifth day, but after 10-15 days when we clean up, we do that. So all these are going and adding up to the load, to antibiotic load, to the water, and finally to the soil. So I think if we can stop, minimize taking, and then if we are taking also, how are we taking care of the disposal at our individual level before anything comes up on a very strong policy and regulation basis? Can we help it at the individual level? That I think will try to address some of the things in terms of drug resistance coming up. Am I?
Okay, thank you.
Manasi?
Yes, ma'am.
So,
I think
madam,
there is
we have
Namrata.
yes, yes, Dr. Namrata Ray is asking, Are the MIPs designed to capture the antibiotics like an enzyme substrate complex? So it should have a pocket that may react. And once they are separated using the magnetic core after one cycle, how are they made free to be used again?
Yeah. So should I go back? I think I'll go back to the slide and explain. That will help. Yeah. So you can see this, right? So
Yes,
it
ma'am.
is not, first of all, it is not an enzyme substrate reaction at all. Okay. So what is it then? It is basically, if you see here, they are synthetic things. They are, first of all, they are not biological at all. Second thing, it is not enzyme substrate reaction. What is it then? So if you see here, we tried creating something similar to a cage. So what is this cage all about? So this is, suppose this is ciprofloxacin. Okay, so fluoxacin. So this is one of the antibiotic, and I want to capture this antibiotic. Okay, so where can I capture it? So I can capture it at a place where it exactly can occupy the same structure, I will say. So this is having a particular structure, right? So if you see about, if you see these, yeah, we had some, the structure of antibiotics also, I think we had like amoxicillin and everything. So yeah, so this, suppose this ciprofloxacin has a different structure which is very different from amoxicillin. If you see here, so there are two rings and all these things. I'm not going to get into the chemistry. I'm rather talking about the physical space which this molecule is occupying. So this physical space occupied by tetra, physical space occupied by amoxicillin, physical space occupied by ciprofloxacin is different. All of them have a different physical space. Now, if you, what we are trying to do is we are trying to exploit their physical space. Okay. So their unique physical space, we are trying to, we created a cage here. And how did we create the cage? That is because we have the monomers which made a polymer around this. So if you see here, this is, suppose this is a island. So this molecule is an island, and against that we have created a polymeric layer. Okay. Now this polymeric layer is against what? Is against the exact shape of this, of a particular antibiotic. So after we did this, what we do? We take out that antibiotic. So against the antibiotic, we created the polymer. And then when we take out the antibiotic, what is left is what? Is an empty space. So you have taken out the template, you have removed it. So this white space is an empty space. And to what does this empty space correspond? It corresponds exactly to the antibiotic against which you made the polymer go around it. Okay, so each molecule you use, suppose it is tetra, so this space will be specific to tetra because against tetra's physical space you used this particular polymer, and you rather carried out the polymerization. Okay, so now this is what making it very specific. So every time you allow it to interact, it will so basically what is there in the wastewater, there is antibiotic, right? And we allow this magnetic MIPs to be there in wastewater. So what will happen? Each of these antibiotics will come, and suppose you have 10 MIPs. So in 10 MIPs, 10 antibiotics will come, and if you have a mixture of 3 out of that, only if it is made for tetra, only tetra will come and bind here. So all 10 tetra molecules will come and bind to 10 tetra MIPs. And after
Right,
that,
ma'am.
as I told, we can remove it, and then you can get it, and you can dissolve it, what we call, through methanol, and then you can take a simple spectrophotometric reading, and each one of them will have their own lambda max, so we can go for it, and it will be very specific to it. So could I answer your question, or there is some more part of it?
No, ma'am. That's what my the question I actually wanted to ask was what I meant by the enzyme substrate complex. I know it's not biological, and it's just an analogy to say that it has a pocket which will
Yes,
fit the particular antibiotic.
yes,
And the other thing I wanted to know was when the antibiotic is fitting in that space, whether the interaction is chemical in nature or is it just a physical fitting thing. Because if it is a chemical
Yes,
interaction
yes.
by
It's a physical adsorption. That is why if you see at some point we have used the
right,
word desorption. It's adsorption and then desorption. It's
right,
getting
because then
absorbed
that
because of the physical space.
right, so that is why it will be easy to free it also.
Yes,
So
it
that's
will
what
be
that's
dissolved.
what
That is why it is dissolved. And MIPs are, widely used in sensing for many different things also. We have tried using it for antibiotics, but it is used widely used in sensor development and all. So it's quite used.
right. Okay, thank you, ma'am.
Thank you.
Okay, okay, ma'am. We have a question from Kaushik Sharkar, Dr. Kaushik Sharkar. He is asking that he has got a question that in modern science, instead of antibiotic, mild steroid also is used for treatment. Is it safe? He's just asking it out of topic and just out of curiosity.
Yeah, yeah. So steroids are being, used. We know that, and that is one of the last resorts when we talk about treatment of many things actually. So safety aspect is rather than safety, it's a requirement. Nothing works, so we are going for steroids. So that is one of the things I would say. I'm not a medical practitioner, but what I understand is it becomes more of a requirement rather than safety issues. Safety becomes secondary there. There becomes survival issues are prioritized there. So that is what I can just say.
Okay,
Okay,
ma'am.
ma'am. Thank you.
So
Yeah,
thank you,
Kaushik has
Dr.
some more. Yeah,
Sharkar.
Kaushik, tell me.
So for removing, we have another question from Shujata Datta. She's asking for removing antibiotic contamination from drinking water. Is RO filtration technique sufficient enough?
See, the filtration part for if the filtration part does to some extent address this depending on the membrane which is being used, but it doesn't address it fully, and that is why even when we check drinking water, and I think these studies are coming up, even drinking water supply drinking water contains antibiotics, so it does not take care of it fully. That's what I want to say. In fact, temperature can take, I think, sometime before someone was talking about temperature and all, right? So temperature to some extent takes care because especially when they are released into water, because some antibiotics are thermolabile, so they cannot withstand high temperature. So in that case, they will break down, but they will again break down to what products we don't know if the products are more toxic. So we don't know about that. So but temperature does something to antibiotic stability in terms of stability, chemical stability.
So, ma'am, we have another question from Abhijit Biswas. He's asking that if he finishes one course of antibiotics, can another course of antibiotics from a different group start immediately, or there are chances of side effects because of this?
No, no, no. See, each drug entity we take, even if it is an antibiotic or something else. So when the doctor prescribes, they definitely they take care of what you are speaking about. But antibiotics have their own mode of action. So generally, they do not interfere in each other's mode of action. That is why, see, penicillin works in a different way, and tetra will work in a different. So they have their own mode of action. So generally, they will not interfere, I believe. And of course, the doctor definitely takes care of this when they prescribe you. When they prescribe you drugs, they definitely take care of the combinatorial effect which is going to happen.
Okay, so any other questions we have? Dr. Arunima Datta has a question. Please proceed.
Thank you, madam. Ma'am, I wanted to know that you just now you showed that how the molecular caging happens and the antibiotic molecules are being taken care of in polluted water, but how to quantify how much of which antibody is there in the polluted water? How to quantify that?
Yeah,
Are
yeah.
we
So
going
that's
to
what
mix
I think I already
arbitrarily
showed.
this
I'll go
number
back to
of
my slides.
molecules of MID
Yeah,
to
yeah, sure. I'll go back to that. Yeah. So this one, which you can see here, yeah. So this is all about this. This thing helps in capturing because we are allowing the wastewater and the MIPs to interact. Okay. So this is around a 30 minutes retention time. So it will interact. And after that, what we allow is what we do is we allow the water to flow through. If you open this, the water will go away and all these MIPs will be collected here. So once it is collected here, what we do is we pour methanol, as you see here, as a desorption solvent. And now what will happen is the antibiotics will be dissolved out and it will be in the methanol. Okay, so this methanol containing antibiotic, what we do is we do a spectrophotometric analysis in the UV absorption range, and each antibiotic will have its own lambda max. And from that UV absorption, here is what we have quantified. Okay, to what extent? So this is a linear range. So basically, it's an UV absorption based study after you get the antibiotic, because the challenge was to get the antibiotic from the wastewater, because you cannot take wastewater as such directly for and spectrophotometric study. So that is why all this process of uh, taking out the precipitating out the antibiotic from the wastewater and then go for a spectrophotometric analysis, which quantitates and tells you how much antibiotic you have in the wastewater. Could I answer your
And
question?
yes, ma'am. And another question is that after knowing the quantity, how much quantity of this antibiotic capturing material we need, isn't it a bit of difficult to add a huge amount of these particles in a pond or pool, or where
No,
the
no, no,
in a
it
tank?
cannot be. Yeah, you are right. You are basically talking about what will be the operational mode, right? Yeah,
Yes,
doing it in lab is okay, but what will be the operational mode? Yeah, true.
yes.
So it cannot be done that way. It has to be integrated with STPs or treatment plants where it is taken. We cannot. That is why we are immobilizing also. It cannot be released it into any water. Okay, this has to be integrated with the treatment plants wherein the water is treated and then it goes and then it is supplied and all. So it has to be done in a fixed position in integrated with the treatment plant.
And how much will be the cost of this treatment in the treatment plants?
So
Because
this, if we have not done any scale up studies as of now, but we can definitely comment on the production of the MIPs. And as you see, as if you see the going towards the economy, if you see that we used monomers, right? Monomers which are quite acrylamide and all, which are quite uh, uh, they're quite common monomers. So those monomers and then their synthesis, all this process taken into account, it's not a huge costly process as of now. But if you want to really comment on the economy of the process, then it has to be the scale of things has to be taken out. Like right now, what you have commented is what are the material being used, what is the time taken, so that we talk about the process time also, process time and the entire process we have talked about. But the actual comment regarding the economy can be talked about only when we see the scale up in terms of how many liters of water per hour can be remediated and such things. So exact costing we cannot talk about right now. That can be done only after the scaling up.
and only we are here only talking about 3 antibiotics as such, but there are
Yeah,
a
yeah.
huge number that
Yeah,
is
there are a
left. So,
huge number. There are not just there are a huge number. Many of them are actually also not stable. So if you see, these are the most used, widely used ones which we have chosen because of their, they were the highest amount which were present in Indian population, and there are more, of course, in the list. There are more on the list.
ufloxacin nowadays is being used rampantly, that we can see.
Yes, yes,
Ufloxacin.
yes, yes. So that was the highest one actually, which was found at the highest level in the Indian population. Different population actually has, if you see some Chinese studies, they have a different antibiotic use pattern. Definitely the disease varies, and the availability of antibiotics and many other things play a role. So different parts of the world have a different list of antibiotics, which is amongst their environment.
And maybe after this COVID scenario, we are using more of this cetirizine and all
Yes,
kind of
yes,
anti.
yes. Yeah,
More
many things actually. We are
okay.
rampantly using antibiotics, we should say. We really don't want to. We just at the first go only. We all of us go to antibiotics.
Thank you, madam. Thank you.
Thank you, Dr. Datta. Now, Prajit Nambiar is asking, won't activated carbon treatment remove antibiotics from water?
Well, adsorption based things, if you talk about the existing ones which we compared, no, they do leave remnants to the extent that all of it doesn't go away. And that is why there was a need for, when you talk about treatment, when you talk about treatment, the limit of antibiotics which is left after the major remediation things which are available, like if you talk about common treatment plants, if you talk about a common treatment plant, the way they take care of things, lots of remnants are still left out. But activated charcoal as such has never been, I mean, it has not been a part of treatment plant. But if you talk about using it as a material which can adsorb, it will definitely, I believe, adsorb to some extent. But the adsorption is probably what we have not directly done any studies on that, but through literature and all, whatever we could gather, because that is why when we did the literature and search and went ahead with the study, we did this. So it does not remove all of them, and to the limit that it is not toxic to the environment.
So, any other questions we have? So, I think we don't have any other questions, right? So, I think there are no other questions, but again, the number of questions actually brought to the front the relevance of your lecture and your lucid way of presentation, ma'am, that actually could ignite so many questions in a session. So, to wrap up the session, basically, we are extremely thankful to Professor Sanjukta Patro for delivering such a wonderful lecture on sustainable solutions for monitoring of antibiotics and assessment of water health, from giving stress to the crisis of water, and again to bring about she talked about how we could actually look into disposal. We could actually build in a collaborative effort from the individual side and the collective side, so that we can actually better deal with this problem of water stress, which includes, of course, the use and abuse of antibiotics within water as it gets disposed. So we are extremely thankful to ma'am. I would also take the opportunity of thanking all the participants for your question, and of course to ma'am for answering, for being patient enough and being with us and answering all the queries. So with this, we will break for lunch. Today we have a bit longer lunch session. We will meet at 2 with our 27th lecture for this day, and we will be meeting for the lecture by Sri Kunal Vora. So with this, I thank everyone, and I call this session a close. Thank you.
Thank you, Titasha, and thank you to the thanks to
Welcome,
the
ma'am.
organizers for inviting me, and thank you, audience. Thank you.
Thank you, ma'am.